Good afternoon, everyone present here. I am Sharanya Dumbre Patil and I will be your host. I'm honored to extend a warm welcome on behalf of Amity University Mumbai to this distinguished lecture series on biology of tuberculosis. Today's lecture will be delivered by esteemed speaker Dr. Anand Krishnan, and the topic is Emerging Role of Exosomes in Tuberculosis. This series is organized by Amity Institute of Biotechnology. As we strive to deepen our understanding of tuberculosis, our attention has shifted to exosomes, a class of small extracellular vesicles. These exosomes have recently garnered attention for their potential involvement in complex interplay between tuberculosis pathogen and the host immune response. Guiding us through this exploration is esteemed expert whose insight promise to shed light on the nuanced function of exosome in context of tuberculosis. We will delve into the molecular mechanism underlying their role and consider how this newfound understanding might contribute to our strategies for diagnosis and treatment. That said, I would request Abhishek sir to give welcome remark.
Thank you, Sharanya. Am I audible? Okay. Dr. Anand and I, we started our PhD journey together at the Urban University of Technology. Let me just give you a brief introduction of Dr. Anand. Dr. Krishnan Anand is a senior lecturer in the Department of Chemical Pathology, Faculty of Health Sciences at the University of Free State, South Africa. He is an internationally recognized researcher working on integration of clinical chemistry, medical biochemistry, bioinformatics, and nanobiotechnology. Dr. Anand leads the Precision Medicine Integrative Nanodiagnostic Laboratory, which is currently engaged in biological functions of extracellular vesicles and development of nanosensors in brain biopsy analysis and non-invasive disease diagnosis. Dr. Anand served as senior researcher at the Durban University of Technology, University of KwaZulu-Natal. Dr. Anand is a rated scientist by NRF of South Africa. Dr. Anand has received several research grants, the most significant being a research grant from the Swiss National Science Foundation. Dr. Anand has 135 research publications. He has authored books, chapters. He has also edited 7 books. He also has several patents for his work. He has successfully supervised and mentored doctoral students and also hosted postdoctoral research fellows. As a researcher, Dr. Anand has received various awards in academia, such as the Best Postdoctoral Research Award, Young Scientist Award from Pearl Foundation for Educational Excellence in India, and Top Publisher Award. Dr. Anand is also member of several distinguished societies, such as American Chemical Society, South African Chemical Institute, Professional Chemistry Membership, and Royal Chemical Society, and the International Society for Extracellular Vesicles. So, I welcome Dr. Anand, and we are eager to hear from you. And I'm sorry about the technical delay.
So we're done now.
Yes, you can
Can
start.
can you start now?
Yes.
Okay. There's a lot of students joining now, so I'm admitting everyone. So side by side. Okay. Welcome to everyone. Thanks for inviting me to giving a wonderful talk with the Amity University. Professor, thanks for Professor Abhishek Kuldev inviting me to give a lecture on tuberculosis. Is a wonderful topic. Yeah, really appreciate your invitation. I like to give good presentation with your institution. Yeah, basically I'm from University of the Free State. I'm working with the Department of Chemical Pathology as a senior lecturer in clinical pathology. My research focus on more into liquid biomarkers, liquid biopsy, like metabolics and proteomics. There's so many things in liquid biomarkers, circulating liquid biomarkers. Especially I'm working with the field called extracellular vehicle. It's a trending research going on with the extracellular vehicle. It's also called exosomes. So my field, I'm trying to link with the tuberculosis because basically I'm working with the more into cancer biology, but it's very interesting to know the tuberculosis is also very, the exosome and the tuberculosis combination also very interesting research field. So I'm going to give some basic talk about what is the exosome and what is the tuberculosis current scenario, and how exosome extracellular vehicle we can link with the tuberculosis research. So far, what research going on, and then what we can do in future. Even from this lecture, researcher or students, you can explore a new field from this extracellular vehicle with the tuberculosis. Okay, so today I'm going to talk about emerging role of exosome in tuberculosis. You can see my slide, front slide, the patient with the TB. They're pulling blood from the patient, then they're diagnosing through different analytical technique. They try to find out new biomarkers, then they try to develop a medicine or monitoring patient. So this is this whole step-by-step method they're doing for a tuberculosis patient. Okay, now, yeah, Abhay, this next slide. This is basic things like epidemiology, the global threat, emergency of residents, diagnostic and challenge, like a phenotyping diagnostic, molecular diagnostic challenge, the emergency tool and method. Yeah, you can go next slide. Yeah, this is the just tuber epidemiology, how this tuber closis is spreading through one hosting to another one host. So you can see oldest is a world oldest human pathogen. It's called a mycobacterium tuberculosis. It's a transmission through the droplet containing the pathogen, like sneezing and hopping, is spreading to one host to pathogen to another one host. Transmission rate usually happen 2 to 3 cell at the time. The growth and the spread multiple once every 24 hours. Yeah, these are the basic about how this tuberculosis bacterium spreading. Yeah, you can go next slide. Yeah. Yeah, tuberculosis most successful human pathogen. Why they're saying most successful tuberculosis pathogen? Because still we're struggling to wind up this the pathogen, because it's not like other pathogen, like easily some antibody can cure this pathogen. Is this pathogen itself modified the genetic mutation based on, like if you even if you take drug or you monitor if you go for a medicine, is it updating their genetic mutation is upgrading, but still we need to find out right diagnostic and right treatment to cure this pathogen. I mean, destroy this pathogen. That's why they're saying most successful pathogen like that. Over the 1 million deaths past 200 years, biggest infectious disease killer. Why so successful? Very slowly grow rate on the latency, activation, and reversion. More recent reason, HIV epidemic and drug multi-drug residents, breakdown national and control program, migration from one place to another place. Like so these are the 4 points very important for this pathogen successful HIV epidemic. What is a HIV epidemic? The TB and the HIV always linking together. Okay, drug and multi-drug residents. Whatever drug we developing for the TB, still this is this pathogen getting residents. Okay. Also break down the national control program means this is this pathogen, population affecting. So we plan the whole program. I mean, a national control program. Medical point of view, there is a breakdown. Okay. Yeah. Go for next slide. Yeah, this is just overview for 2016 status. How the TP cases increasing. So every year increasing. So you can see susceptible testing growth based up to 2 months, molecular fast but consequential, and these are the two drug, isoniazid and rifamycin is a currently MDRP drug. Then only 54% are successful treated. So just is overview of TB rate actually. You can go next slide. Yeah, these are the current diagnostic TB, but I will you can see it's not clear, but I will explain what are the diagnostic. There's so many diagnostic kits is there. Molecular point of view is there. See, screening point of diagnostic and treatment point of diagnostic and monitoring point of diagnostic. There are different type of diagnostic method is there. The screening diagnostic just lateral flow assay method or some basic test kits method. But diagnostic treatment monitoring, drug monitoring, when a patient admitted in hospital, they're giving medication, then they're more following their recover from the TB. So these are different diagnostic steps is there. I will explain following slide what are the diagnostics there. You can go next slide. Yeah, molecular diagnostics. See, these are phenotype. Actually, what are the gene expressing based on the drug residence TB strain actually. So for isoniazid reflow, these are the drug for TB, but still, even though the drug is there, but is getting more mutation and more residence on this drug. So based on this genetic mutation, we can find out what are the strain is there. There are different type of strain. So we can, based on this mutation, we can find out what type of strain is there. So molecular diagnostic, molecular point of view, like a genetic point of view, RNA, DNA mutation, we can find out what strain every single every patient have. Even country to country, the strain will change based on the genetic mutation. Okay, so molecular diagnostic very important for to find out what type of TB strain in the patient. Okay, go for next slide. Okay, these are the challenge and the emergency tool. We need to find out alternative medicine for, if we know the residence mechanism, we can find out alternative medicine. So we need to find out the whole genome sequence for that pathogen, TB pathogen, but still now we're not finding the whole genomics because every strain have different genomics. So we need to find out the whole genomics first. Based on the whole genomics, we can develop a new therapy. Then another one, minor variant calling is, but yeah, you can see if the different phenotype and the genotype we is giving we need to find out the gene sequence for the different type of pathogen. So we need to we can develop a drug molecule for that one. Okay, here you can see epigenetic point of view. So some techniques have mutation or DNA, some DNA methylation. What happened with the molecular point of view, we can find out through some techniques like that's called SMRT, scene second techniques. So from this slide, it's a more into genomic scene sequence actually. If you develop genomic scene sequence techniques, this field, this tuberculosis field, we can find out the exact mutation from the drug resident tuberculosis bacterium. Okay, so if you find the way to how to find out the mutation of drug residents tuber pathogen, then based on that one we can develop new therapy instead of sitting with a traditional drug therapy. We can move for a next generation drug therapy. Okay, go for next slide. Next slide. No, you're going front. Next one. Not this one. Next one. Yeah, next one. Yes. Yeah, whole genome sequence for that challenge. Actually, if you want to develop a drug or therapeutic, we need to find out the whole genomics. So you can see this is a basic about this slide will explain about basic about current genomic sequence from the tuberculosis pathogen. Okay, the genome size is a 4.4 MB, and 40% of unnoted genetic, dark matter. Still, we need to find out a lot of genetic mutation. Type 2 genome, many large and small repeats. Circular unbalanced genome, GC rich genome. You can see these are the content. And the GC BIOPS platform, that PCR amplification, ion tolerant Illumina. These are the technique they're using to predict the genomics. Cultural preparation and the DNA extracts take very long time. Not ideal for scene sequence for the cheaper run in the economic of scale. Okay, so these are the challenge they're talking about when you're working with the genome sequence. The genome sequence is not an easy process. Okay, there is a long process because we need to go for genome sequence. We need to isolate that biomolecules from the tuberculosis. Except biomolecule means DNA, RNA. We need to go for PCR. Then we need to go for PCR techniques, different type of PCR techniques. But end of the time is a long process to predict that genome sequence. So this is not affordable, but it's a very accuratable. But still, we need to find out a more affordable technique to find out the whole genome sequence. Okay, you can find out some certain strain tuberculosis, not for all the tuberculosis strain. For example, Africa have different strain, India have different strain, Europe have different strain. So, but every people have different strain. It's not like everything same strain like that. So if you finding all the genomes in one box is not easy. So this is the challenges they currently they're facing. That's why people are doing research with the try to find out easy way to find out the genome. In meantime, this tuberculosis is multiplicating, developing, is became mutated. So it's not uncontrolled. That's why it's called epidemiology. It's falling under the epidemiology. Okay, this uncontrolled. One side, scientists are working to develop new techniques to predict the scene sequence. In meantime, there's a pathogen also getting mutated, changing the strain, changing is moving forward. Okay, that's why it's unbalanced between the balance between scientific research and genomics mutation. Okay, yeah, you can go next slide. Yeah, this is the emergency solutions SMR sequence nanopore. This is one type of techniques to find out methylation. Is this technique suitable more into find out genetic mutation? Okay, so maybe is DNA methylation in tuberculosis. So just this is one technique they're using. Okay, go to next slide. Yeah, you can see this is a somewhat seen sequence and Dino assembly. Assembly, circulation, constant polishing, methylation detection, assembly QC. This is the flow of finding genetic mutation. This you can find out like a one how can I say this is one type of method to predict methylation, DNA methylation. Okay, so from this one you can predict methylation from the SM scene sequence. Okay, go to next slide. Yeah, this is isonized drug residence bacterium, mycobacterium tuberculosis. So just I'm saying this is a drug residence mycobacterium tuberculosis. Okay, so based on this mutation, we can find out this drug mutated tuberculosis. So we need to find out alternative medicine for this resident bacteria, actually mycobacterium, because this isoniazid is a drug molecule. When a treating them, it got mutation, it can overcome this drug molecule. So we need to, it's teaching us to develop a new drug molecule. Okay, go to next one. Next one. Yeah, pyrazinamide. This is also one of the TB drug currently in the pipeline with the patient. So this is also got mutated with new residence bacteria, bacterium, tuberculosis bacterium. This is also, that mycobacterium tuberculosis is overcoming this drug molecule. So what we can learn from this thing, whatever we developing drug molecule, that bacterium genetic getting residence on this drug. Okay, so how we know, how we can find out? Based on the scene sequence, there's some techniques we can find out. These are the mutated residents still, overcoming their drug molecule. Okay. Okay, go for next slide. Okay, what we have learned so far. Okay, from this thing that pathogen teaching us is not controllable. When whatever we develop a drug molecule for TB, TB variant getting overcome with the drug molecule. Okay, so we need to find out alternative the drug therapy for this tuberculosis bacterium. So this means so far we not finding proper genetic variant, genetic code, genetic sequence from this pathogen. That's why we cannot develop a new therapy. I mean, develop a proper therapy to cure this the tuberculosis. Then what is the meaning of this thing? We're not finding the whole genomics. If we don't know whole genomic of this variant, this bacterium tuberculosis, we cannot develop any drug molecules. So we need to find out the whole genomics detail with a different variant, different residence bacterium. Then only we can develop accuracy diagnostic method. If you find accuracy diagnostic method, then we can go for the accuracy therapeutic method. So this is the so far we learned from this pathogen. Okay, because if we're developing for one genomic point of view, but it got mutating, it got residence with the drug molecule, then it became another one type. So better we need to understand the whole genomics properly, then only we can develop a new therapy for TB. Okay, so this is the so far we learned this from this species. Okay, go for next slide. Okay, see here. what we have learned so far, current to molecular dynamics act as a selection mechanism, and sensitivity and specificity of molecular device should be accessible annually. Okay. Yeah. So yeah. Uh, so what we learn, whatever we do diagnostic is all after the cut is resident, after is that tuberculosis is developed, then only we finding. Before we cannot find, because this is the lack of diagnostic failure actually. If you want to develop, if you want to control the infection disease, we need to predict the earliest possible. Then only we can develop new therapeutic to control that infection. But we are always after infection only we developing diagnostic, we developing therapeutic. For example, take it as a corona virus. So if we know before corona genetic mutation, if we know corona virus, all the detail before it's coming to pandemic, we can develop drug molecule, vaccine, whatever. But if we don't know anything about before, then surely it will come to infect people. Then only we isolating strain, then we finding the genetic sequence, then we developing a vaccine or a drug molecule for that. So these are the lack of diagnostic to predict the earliest possible how it can get mutated, how it going to mutated, how in future it will get changes. So we need to predict before that. Okay, so go for next slide. Yeah, this slide you can see in South Africa and India, because I need to compare with the South Africa and India. Actually, this slide a little bit different from what I discussed before. Before only we discussed about tuberculosis, how got mutated and how drug residents, lack of genomics knowledge, we still struggling to target proper therapy. But this one, always we need to link tuberculosis with the HIV co-infection, okay? Because South Africa has a big issue with the HIV. People are thinking HIV because of man-made error with the illegal sex, the blood transaction, uh, like that. Is not only that way, okay? Actually, when your immune going down, the HIV sometime even people go illegal sex and um, blood transfusion and other things, they will get it easily TB. Okay. Also, the TB patient also will get easily HIV because they lost the immunity. So it's like, so even not only South Africa, India also same issue actually. So always HIV and TB is always linking together because of confirmation. Okay. So when the TB is come, the patient will get HIV. I mean, if he is a HIV positive, he will get TB. Okay. If you the tuberculosis, the patient, if you get the patient got tuberculosis, if you go for some HIV illegal things, like HIV positive blood transmission, the TB patient will lose that immune power, then he will also will get easily HIV positive. So I'm not saying like a TB patient must will get HIV positive. It's something like if you do, like you go for illegal things, he will get easily HIV when compared to non-TB patient, because it's all about immune competition. Okay, because the TB and HIV always play with the immune cells only. So when compared to other infection disease, virus disease, that TB is a very impact on immune cells. Okay. Okay, go to next slide. Okay, so far I talk about basic about tuberculosis. Okay, TB bacterium, so far. But now I'm coming to my research now. So whatever I discussed in the slide, this is the basic about TB, how this TB so far genomics finding, how they're developing diagnostic, what are the current drug in a TB. Okay, those are the basic lecture, basic knowledge about TB and TB research. Okay, what's going to happen future? How we going to, develop? I mean, how we going to control this thing? Not only TB, the other infection disease. Okay, so current medicine is choosing patient come with the disease, then the physician will diagnostic the patient, then he will give for medicine, they go for medicine. Okay, they go for therapeutic. This is a current medicine, current traditional medicine going on. Yeah. Okay, but future medicine is going to be a personalized diagnosis, personalized medicine. Okay, so the patient come with the disease, we we focus on the patient genomics um, things. Then from the genomics, we can treat that patient separately. Okay, so this is called a personalized medicine. Okay, so it's not like, for example, I also got fever, you also got fever, but you going to one medical doctor, you prefer paracetamol, but you don't know my genetics is different from that you. So we need to develop something personalized uh, treatment method. So this will help future to predict, stop that infection disease or any disease. Yeah, we need to focus on personalized point of view. Go to next slide. Yeah, there are so many type of personalized medicine for different disease model. For example, cancer, uh, diabetes, and Alzheimer. But tuberculosis also recently they started precision medicine for tuberculosis. Okay, so tuberculosis, how is working at precision medicine? The main thing first, they try to predict the genotypes. Okay, then based on the genotype they try to develop drug molecules based on the immune suppression, the immune level. They try to develop a new therapeutic method, like currently immunotherapy is playing important role. So we need to find out the immune-based biomarker through that genetic variant. Then we try to give the treatment. We need to monitor the patient how far is reacting. Then we need to decide that how safe the patient. So this is so all about precision, all about how you treating the patient. It's not like all the TP patient have same thing. Every TP patient have different immune level. So based on that immune level, we need to treat the patient. Okay, my immune level different from your level. So based on my immune level, I need to get treatment. Based on your immune level, you have to get a treatment. It's not like every immune level same. That's why when a corona came, some people died for corona because of their immune level is low. Some people didn't die. Like people are we didn't die because we compromise with the immune level. Our immune system reacting with the coronavirus, it against that virus, it got stopped. But some people look healthy, but their immune level is less. That's why some people dead by corona. Same thing. Every human have different immune level. That immune level will stop that any infection disease. So we need to find out to how how much immune level for our individual. So we need to go for some precision diagnostic method. How much immune cells really active? What are the immune cell inflammatory is a biomarker, inflammatory, pro-inflammatory, how infecting. So we need to find out all the things. So that is way of precision treatment. So that will help you to stop any infection disease. Okay. Go to next slide. Yeah, this is the recent trend, how precision nanotechnology to diagnosing MTP. Okay, so these are the, you can see just year-based spectrum of how they are developing diagnostic method in 1982 to now, how more accurate and how the technique developed from the one stage to another stage. So this is, for example, first they start from microscopy, then just radiotherapy and IgRA test, then culture test, then lateral flow test, and genetic test. Now microfluid tests. So these are the evaluation of technology. Okay, to predict more accurately what the MTP, what type of genetic update with the MTP. Okay, go to next slide. Yeah, this is something related to traditional treatment with the nanosystem treatment. Okay, so the one more thing, not only diagnostic point of view, the therapeutic point of view, we need to focus on how more efficient and low toxicity drug observing and drug developing method through nanotechnology, or it's not like nanotechnology, something like we need to develop new method, a new way of to treat uptaking therapeutic molecule from patient for patient. So conventional method, we just we choosing that the patient, we giving tablet, but the tablet is that treatment of some people getting drug satiety, drug toxicity, long duration treatment. Patient don't take the medication, because of the side effect. Also the drug will that bacteria residence with the drug molecule, also low availability of the drug molecule target. So it's not like a receptor target therapy, just taking medicine to cure, but it's not accurate target therapy. So these are the conventional therapeutic, current therapeutic. Okay, but maybe in future the drug nano formulation will help to target accurate therapeutic way. For example, improve that compliance, target delivery, for no need to take high dose drugs. We can load dose for proper delivery efficacy, reduce the drug degradation, less side effect, increase the viability of scope. Okay, so based on the now formulation, so we can develop, improve that delivery and the targeting for therapeutic point of view. Okay, okay, go to next slide. Okay, now is the main slide here. This is my research. Pathological role of exosome. Now we came to core of the presentation. So exosome. So see, so far we're discussing about tuberculosis, how the tuberculosis evaluating, then how we need to improve through nanotech. So far we talk about P and precision medicine, how to develop the precision medicine accurately to treat the patient, accurately find out the biomarkers for TP. So we discuss about these things. But now the exosome is a very important role here. Okay, because this exosome is a biogenous material from cells. It's not like artificial from outside body. It's the inside body. Every cell will produce the exosome. Okay, so now people are doing research with the exocell vehicle, also called exosome. Not only cells, the bacteria also producing exosome. Any, virus also produce the exosome. Human cells, animal cells also producing exosome. So if you start work with the exosome, maybe the exosome can give right way to find out accurate diagnostic, especially the genetic point of view, as well as therapeutic point of view. We can find out right biomarker and right therapeutic through the exosome. So I will explain about following slide what is the exosome, how to use this exosome for tuberculosis. Okay. So you can see this slide. Exosome is a small particle releasing from parents' cells. When the cell getting divided, it is releasing from one cell to it will circulating into our human body. Then it will target at the other cells. So the exosome carry some genetic matter like RNA, DNA, protein to one cell to another cell. Okay. Okay. Next slide. Next slide, Abhay. Yeah, you can see this is the original picture of cell, how the cell releasing the exosome. You can see the exosome particles, the cell membrane. This is the original TEM image of exosome. Okay, go back, go to next slide. Yeah, you can see one video. Click that video, Dr. Abhay, so that people can see. Is there any option is to click? You can see this video will explain how the exosome communicating with the cell to cell. This is the original picture of cell to cell communicating with the exosome. You can see the cell, the small particle moving to one cell to another cell. This is the exosome actually. You can then means if the exosome has something is there. If we capture the exosome, if we study exosome properly, then we can find out right diagnostic method, right therapeutic for this any infection disease. It's not only tuberculosis, any infection disease. Okay, so that's why I like to focus on the exosome, because this is involving cell to cell communication. So okay, yeah, Abhay, go to next slide. Next slide. It's fine.
Can you see the slide?
Yeah, it's finished now.
No, can
Next
you see
slide. No. Only I can see only video slide. Yeah, this slide, this is the biological composition of exosome. Okay, so it's not something exosome, small spherical particles and nothing inside, just traveling here and there like that. The exosome carry a lot of genetic matter. Okay, it have protein, it have RNA, it have nucleus, it have all the growth factor, it have RNA, DNA, nucleic acid, it have lipid, it have some signaling protein. So it look like all the whatever cell parent cells is there in this exosome carry all the genetic matter to another one cell. So it is like a grandfather to father, father to son, like that. So if we capture this exosome, if we study this exosome carefully, we can develop new genomics pathway, then we can develop new therapeutic, because these are the lack of experience with the researcher. I mean, finding accurate genetic mutation and genomics. That's why we still struggling to success that therapeutic with the TP. Okay, so these are the biological composition of exosome. So whenever that biological combination talking about nucleic acid, protein, RNA, DNA, then means something is there. We need to find out from that place. It's not only we can focus on cells releasing material. Cell have nucleus, cell we can isolate RNA, DNA from the, we can develop genomics. We need to find out that cell derived material also. So it's not like the two things I want to tell you. Okay, maybe this exosome carry that disease biomarker to one cell to another cell. But if you focus on this exosome, if you isolate this exosome, capturing, studying this exosome through proper way, we can develop a new uh, diagnostic method and a new therapeutic method for any disease. Okay, so okay, go to next slide. Yeah, now you can see this is the clear picture of how microtuberculosis involving the exosome production. Actually, you can see the green, there's a bacterium is going to cells, okay, lungs cells. Okay, so the exosome, biogenesis exosome, as already I discussed before, exosome release from cells. Maybe this microbacterium you know releasing their biomaterial to cell, the cell will release that exosome. That exosome have genetic material like RNA, DNA, protein. Okay, that one can react with other normal cell, maybe is no, transporting that TB biomarker to other cells, TB genetic code to other cells. So that's why you're getting infected. So if you capture this exosome, that TB impacted exosome, if you study this exosome, maybe we can find out proper therapeutic method. Okay. Okay, go to next slide. Yeah, this slide for cancer medicine, but still we need to focus on TB. Okay, so this currently cancer oncology field, cancer treatment nowadays they're going for personalized treatment. So what you can see this from slide, this is the patient now. The patient have, for example, if you have cancer like prostate cancer or blood cancer, we can take the patient sample, we can isolate the exosome, we can study the exosome genetic biomarker and whole genomics through the exosome. Based on that genetic information, we can develop therapeutic like immunotherapy or chemotherapy or radiotherapy. Then we can treat the patient. Okay, so this is the personalized medicine for cancer. Same thing in future, we can go for tuberculosis also. When a patient have TB, we can isolate the exosome from the patient. We can study the genetic mutation from the exosome. Then based on the genetic mutation information, we can give proper therapeutic for patient. Okay, so this is the treatment point of view. The another one point of view, from the exosome, we can find out that particular TB strain, how producing the exosome, what genetic matter releasing to that cells, that exosome, so we can predict easily. So there's a lot of research we need to done. Okay, so for cancer only they're focusing so far, but still tuberculosis we need to do. Maybe some other research group they're doing research with the tuberculosis. Okay, but what I'm trying to say, exosome can give good information when compared to other biomarker, other sample like currently blood sample, urine sample, serum, saliva, they using for diagnostic, but that is out of cells, circulating biomarker. But exosome not like that. Exosome coming from core of the cells, so we can find out the accurate biomarker from the uh, exosome. Okay, go for next slide. Okay, you can see clearly this figure for TP, how they're using the exosome. Okay, You can see exosome isolation. This is the patient study. Drug resident TB, the 26 patient and a healthy patient, 16 patient. The isolating serum, from the serum, the isolating exosome. Then that after isolating the exosome, they studying Western blot, some biomarker characterizing the exosome. Then isolating RNA from the exosome. Then they going for a frame design. They developing, scene sequence model. Then they going for treatment. Okay, so this is the method we need to follow. People are currently doing research with how they are using exosome for infection disease like TB and HIV like that. But already cancer is already established. So in the future, like a precision medicine, exosomal point of view, precision medicine will help to cure that patient. Okay. Okay, go to next slide. Okay, so this is the slide. You can see
Dr.
like
Anand,
how they can develop
hello,
precision
can you hear
medicine.
me?
Yes,
Dr. Anand?
yes,
So, Dr. Anand, we have 5 minutes more. Okay.
yeah. This is last slide actually.
Oh,
Yeah. So you can see the patient, then isolating cell from the patient, then we culturing the cells from the patient, then we developing therapeutic, isolating exosome from the cells, then we treating the patient again, same patient. So look like a self to self treatment like that. So we can understand easily genetic variation from the patient. Then we can develop, we can identify, we can understand about immune response to the patient. Based on that one, we can treat again the patient. So this is the mechanism we can develop in future based on exosomal biomarkers. Okay, so this is called precision medicine. So precision medicine, we can is nothing but we're going to monitor the patient immune system, patient disease stage level. Based on that one, we're going to develop a therapeutic method for the same patient. So it's not like something common drug for all the patient. It's a precision medicine drug for the particular patient. Okay, so this is future going to come up. So TB, like a infection disease like a TB and a HIV and a cancer, we can apply these techniques to control that disease. Okay, so this is my reference. Okay, thank you for. I'm done with my presentation now. If you have any question, you can ask me. I'm welcome to answer any question. Yeah.
Saranya.
Since
Just I have a question. Can I ask?
Yes.
Yeah, this is Dr. Shashank. Exosomes can be released by both bacteria and host cells. So when MTB is outside the host cell and when MTB goes inside the host cell during infection, what changes do you expect in the exosome composition and also in the frequency of production? And will that
Maybe.
exosomes can act as a kind of interaction mediator from the bacteria to the host during infection?
Yes, that's why that exosome act as a, maybe it will carry that MTP genetic matter. It carry to one cell to another cell.
Mm.
So that bacterium, it will change that cell atmosphere. So that's why you can see that the slide. The bacterium carries some genetic things. That genetic things releasing to cell. That cell when you're getting dividing, the cell getting life finishing. Exosome carry all the genetic matter from that MTP infected cells. It will release. It go to another one cells. That's a mechanism is still not clear. Those people are finding. That's why it's not only TB. All the disease, most of the disease, the exosome playing important role to communication, cell to cell communication. So if we capture that the cell-derived exosome, the infected cell-derived exosome, maybe we can find out accurate genetic biomarker, accurate detail from that infected cells. Then we can is not something like I'm saying genetic. There's so many things there. You can find out your inflammatory level of the cells. You can find out growth factor of the cell. You can find out other other things like immune related biomarker. If we know all the detail through this the exosome, then we can easily can say okay, this is the disease level, we need to treat this way. Because we find a new biomarker, we need to focus on this therapeutic. So the drug company and the drug developers, pharmaceutical, can involve to develop a new therapeutic based on that infected derived exosome. Okay. Yeah. Correct.
Thank you.
Yeah.
Thank you, sir, for such an insightful lecture. I would now invite Dr. Abhishek Kulde, sir, Associate Professor here at AIB, to give vote of thanks.
Thank you, Charanya. Thank you, Dr. Anand, for a wonderful talk.
Okay.
So I extend
Thank
a
you.
hearty vote of thanks to our speaker, Dr. Krishnan Anand, who spared time from his busiest schedule to deliver the talk. Today, we had an opportunity to hear his thoughts about the conventional approaches in diagnostic and treatment of tuberculosis, and also novel approaches like extracellular vesicles for treatment and diagnostic of not only tuberculosis but other diseases. So I'm sure this will encourage our students to consider this area for their future research interest. I would like to extend my sincere thanks to our Honorable Vice Chancellor, sir, for his continual guidance and encouragement to organize such events. I would also like to thank our HOI Inspector Dr. Suprasanna, sir, for his constant support. I would also like to thank all the deans and HOS for their presence for this event. I extend my thanks to faculty and most important our students at AIB for their participation in this lecture series. I'm also thankful to conveners Dr. Shashank for organizing this wonderful talk series, Saranya for helping us with the conducting these talks, and our IT department for smooth conduction of the talk. So thank you everyone. Thank you, Dr. Anand.
Yeah. Thank you, Dr. Abe Kuldev, inviting this lecture session. Yeah. Actually, my advice to student: please read about their XSL vehicle. You will get a lot of opportunity to research with the XSL vehicle. For example, honors and master student, if you do some project with the XSL vehicle, I'm sure you will get a PhD in a developing country like Europe and USA. Because from my knowledge, I can see the lot of big university, big research group focusing on XSL vehicle research. So you people are still young now. So if you do some work with the exosome, like a small project, like your master project or your honors project with this topic, then you put your CV in, you work with the XSL vehicle, then apply for PhD in another country like a big institution, you surely will get PhD. Because recently my colleagues, students, they got a PhD in America, very good in Mexican university because of this project. So also just to read about what is XSL vehicle, how is we can use for our research project. So you can, if you do some small project with this topic, then your CV will get good impact. Then you can search a PhD in UK or Europe or US to get a big institution. Okay, so this is my advice from this my talk. It's not like something I'm sharing my knowledge, research experience. Something I want to share about your, your academic, instead of academic, how to get job, PhD opportunity in other country also. Okay, try to use LinkedIn nicely. Okay. Thanks for inviting me, Professor Abhay Kuldev. We will meet again. Okay. Yeah.
Sure, sure. Thank you very much for your
Thank you.
words of wisdom to direct our
Okay.
students for their future in career.
Yeah.
Thank
Sorry
you
for
very much.
my laptop issue. Next time
No,
is
no,
not going
it's
to
okay.
happen. Yeah. Okay. Thanks. Bye.
Thank you, Dr. Anand. Thank you.
Bye.
I'm
Bye.
ending
All.
the meeting here. Thank you very much.
Okay. Thanks. Bye.